Hello, dear all beautiful learners. Welcome all to our today's session. Once again, I hope so you are getting well and fine, but not as the connecting of the study properly. Since we know the pandemic situation, your college started, but it is online based. So you are getting bored at home. And what about your study? I think so. It is not getting well and fine. What a student deserve for, but actually I hope so, who are always try to connect the study, what was going on the college, what was going on the subject based knowledge to collect, they are getting improved day by day. But who are totally detaching from the study, I think they are not able of getting the terminology, because intermediate level is not as the 9-10 level of science, because it is must and vast upgradation level of your study. So from my side, I advise to you, please, if it is going on through on a specific subject base or any other way, if the topics is going on there, please connect with that class so that you can collect some important information which will be very helpful for your enriching the knowledge. So now today we will go through understand about replication. So replication is very important topics of this chapter 1, and the replication process we already learned from the any previous topics, that is semi-conservative process is the world acceptance of the replication process. But how can it going on there, we will try to understand. Before that, we will try to draw the figure here, so please follow me here to draw the figure. I will draw here in a small form because I need to draw this one of getting like that. Smallest I need to write here of the process is going on there specifically. Okay, I just make it like that. Yes, so in this way I just draw a shape like that. So follow me, my dear students. In this way we will try to understand the process of replication. So here make it double this one, and in this way you will try to draw and make it double this one also, this strand. So we just try to draw a structure like this one, and also having that indicating that parts. Suppose this one is the 3 prime and this part and end is the 5 prime, and this one is since 3, so this one is the 5 prime and this one will be the 3 prime. And in this way there will be a figure or a structure like that. I will draw this way, make it double, and in this way we will ensure this one and make it double. Please use the pencil, because if you don't use the pencil, I will erase after some time, some addition, subtraction, some erasing part when I erase. If you use the pen on just instead of pencil, that times there will be no chance of erasing or just adding something this one. So use the pencil for drawing the figure all time. And yes, we will ensure this one. Okay, so just draw the figure in this way, just follow me in this way. I just draw the figure. What is this, sir? I will explain that one. The complementary strand, what are going on there, we will try to understand here. And if we try to understand in this way, okay, we can erase something and make it half part, half, okay, half part. And we can ensure that this will be the dark part and this strand will be the dark part, almost of course will be the dark part. I use the marker, so you use the pencil. So there the dark part, we will use the dark part is one side, and another side will be the light part, that means no darkish part in there. So this will be in this way, we'll try to draw the figure. And this strand is also, we will make it dark here, okay, and half part, of course, half part, half part, and here I need to draw this one this way for ensuring. I told you I will add something, I will subtract something, so we will try to draw in this way. And also having that part I can erase here for a while, that this will be this one and this will be this one, and some part will be just connecting out from here to know about that. So this one will be as like as that part. So we will try to draw this one, and follow me to draw the procedure, what the procedure is going on there. There will be again similar in the previous time what we draw there. Drawing figure is most prominent part to know about the everything what we actually demand for in that parts. Okay, so we just try to draw and follow that procedure, what's going on there. In this way we will try to draw, and then also we will draw here a structure like this one. Okay, so here also we can try to find out some structure this way, and also in this way we can draw this one, this type of figure. And here the covering, suppose we will try to draw like that. Okay, the half part will be the dark, half part will be the dark. We are near about to complete the figure of drawing. So my dear students, look at here, this is not as like as easy what we think for. Drawing figure can always make you confident to write something about to know about that. So here will be a fork like a structure in the frontal part. That times we try to draw this one, and there also be like that. All the parts will be connecting. We have not enough space of drawing there. Okay, it is not the matter. We'll draw this one in both part. In both case we will try to draw some enzymatic substance, protein SSBP. We already we will try to draw this one. If we draw this one, we will try to mark the figure, and not only marking the figure, we will try to have that specific parts of specific points, like the template. This is the template act as this strand of the DNA. This is the parental DNA. Okay, parental DNA. Okay, template. And later on this one, lowest part of that one also act as the template. Since we know about that template, and this one is also having the parental DNA. And what the part we added here, that is the important. This is at the leading strand. This one act as the leading strand. I write in short form, a small alphabet, so do marking here, dear students. Leading strand, that means daughter DNA. I think you got that one. And lower part is also lagging strand. This part, this one is also known as the lagging strand. Lagging strand. This is also the daughter DNA. So first one we tried to understand. This one is the ligase enzyme. Ligase enzyme. And these are the DNA polymerase enzyme. DNA polymerase enzyme. Then we can say these are termed as the Okazaki strand, or part or segment you can write. Okay, these are called the replication fork. replication fork. These are called the replication fork, and you can see this both part is the SSBP, SSBP, SSBP. Okay, in MCQ or general sense, in short question types of question, they will be asking that what is SSBP. So you will write the single strand binding protein. So these are the SSBP, single strand binding protein, and this one is also act as DNA polymerase. What we actually mind for DNA polymerase. So this one is also important part of knowing that one. And also here in the replication fork, there is some enzyme, that is helicase enzyme, helicase enzyme. So these are the common parts of knowing that the figure. Of course you will mention there the figure replication, the process is going on there, and this process is very important, and this process how going to be understood, I will make you mind, because it is a big process. What is going on there, where the replication will be done there. But specifically I want to tell you, follow my procedure. I will give you the guideline. I will try to make you understand what the process is going on there, and on the basis of that case you will try to resolve yourself. And not only that part, you will note of ensuring of your note in here. So very first time I want to make you understand important things, that is, look at here, this type of figure, it is the parental figure with a strand. This is a there will be the twisted part or winding part, okay, the binding part. But here, from here to there and here to there is no binding part. That means one stays here to make it separate. There will be no binding part of the DNA strand. So some enzyme are working there. We will try to make you understand. And they're also having some another strand can attach with the parental template DNA or complementary strand, or with them also the Okazaki strand or part segment to make it match of that one. So when the polymerase enzyme or like that any kinds of enzyme move forward, the fork will also go forward on the specific part where the place of replication, only untwisted part or unwinding part, also unbinding part, also having the selective part for draw the figure or making the replication. Because I told you in very last class, replication is nothing but the duplication or photocopy of the parental DNA. So another strand can grow inside them when the process is going on formerly and forward. So frontal part or twisted part will be unwinded, and also the later part will be twisted again. So there are many enzymes. This is the process of enzymatic activities. What kinds of enzyme are actually working in that specific part. So very first one I want to make you mind. That first one is helicase enzyme. Okay, so I want to ensure this is the process starting with the enzyme. I will just give you idea about the enzymatic activities, which kind of enzyme working, what types of work. On the basis of that part you will complete your writings, because to know about what kinds of enzyme is going on there. Enzyme, of course, they helps in unbinding the twisted DNA strand. I just tell you this one, and look at here another enzyme process. In a short brief I will tell once again, but I just tell you in which process, where it is going on there, and which specific part you can mention, and on that specific point what types of enzymatic activities. I just write about only enzymatic activities, but this is not all the process. try to understand the class, try to receive me properly, then you can write by your own self with help of the book. That times helicase enzyme helps in unbinding the twisted DNA strand. This is a very important point, that one. Okay. Topoisomerase. Topoisomerase, they're not mentioning there. Isomerase, it helps in not binding again the DNA twist. Okay. Helicase enzyme, helicase not mentioning in the figure, but make it mind here. Similar work done by as the topoisomerase. In case of the prokaryotic cell is can vary out on a specific point, but not mentioning or not thinking about that. Please try to think about. You can write in this way easily with that one. Okay. SSBP, single strand binding protein. So it prohibits to make hydrogen bond of DNA strand again. I will make you once again make you understand. Okay. So and they are act as the template. So what the first one, that is the separation. First point, separation of DNA strand. Separation of DNA strand. That is the important thing of knowing that one. And another one is also be there. Helicase enzyme. Helicase. Oh, already I have written the helicase enzyme. Yes, I already written the helicase enzyme. Yes. So my dear student, the first process of DNA replication, what kinds of enzyme are going on there? Suppose all of the twisted binding part, but we need to do the replication process. So we have to go there with getting to see there. The helicase enzyme helps in unbinding the twisted DNA strand working. So helicase enzyme, make it sure to, yes, no, you don't, you are separate now. No twisting part, you are separate. Helicase enzyme helps in this one. Topoisomerase and helicase enzyme, yes, helicase enzyme separate, but they try to move again, make it twist. But helicase and topoisomerase, no, you are totally separate. They just make it unbinding part, not binding again the DNA twist. So they helps on that case. And we have seen the SSBP on the replication fork. We can see in both other case, they are just what I say, seal, make it seal. So fork will be there, no chance of getting to bind again. It makes hydrogen bond. They try the single strand, they are always trying to make it hydrogen bond with the nitrogen base, again make it twist, no chance, because SSBP working there. So first point, separation of DNA strand. In this way, the many of the enzymatic activities, the strand to be separated. This is very important points also be there. And next point we try to understand. The second one is going on to complementary strand making, or making of complementary strand. There are also some enzyme working there. Also try to understand about that. Making of number 2 step, making of complementary strand. complementary strand, making of complementary strand. So what is what process is going on there in the second page or second case, we will try to understand here. So making of complementary strand, there are some enzyme. I told you the meaning of the way we can write. I just write about the enzymatic activities. You need to know, or you try to understand about that. Okay, so DNA polymerase enzyme working here. DNA polymerase enzyme. This DNA polymerase enzyme always helps to connect with complementary strand, with template, that means the parental strand, and their direction, they always enriching that 5' to 3' end. That means you know that the 5' and 3', this is the 5' and this is the 3'. So their movement, DNA polymerase enzyme try to move forward, and they always helps to connect with this complementary strand and template of DNA. Always try to connect and match them with fixing of the DNA base or a nitrogen base in case of the DNA, and having the connecting part with that specific points of growing 5' to 3' direction for making the duplication here. Okay, so this one is also important thing. There also we will try to understand about RNA primase enzyme also. Primase, they are also working the primase enzyme, and primase enzyme always helps to make it the photocopy of the DNA strand. They helps the photocopy of the DNA strand, okay, with having the primer, okay, primer to help the duplicate, primer help to making duplicate of DNA strand, That means primase enzyme also be there. Since the complementary, we didn't marking here, inside them the lots of enzyme are there, so they always try to make the duplicate of the parental strand, that means the template. So primer, those strand working as to make sure the duplicacy of the parental strand, they are known as the primer. So primer helps to making the duplicate of the DNA strand and move forward with having the nucleotide by nucleotide conjunction, nucleotide conjunction. That means they always try to add the nucleotide, they always try to connect with the nucleotide and forming the nucleotide conjunction, and helps for making the duplicacy or duplicate of DNA strand. So this is also very important thing of primase enzyme here working on done there. Okay, so primase enzyme also having some RNA that this strand can also connect with the RNA. They can produce some small amount of RNA in this case, and DNA polymerase enzyme 3, another DNA polymerase enzyme, finally DNA polymerase enzyme make sure the separation of the RNA strand, which are the complementary strand produced in a small amount. DNA polymerase enzyme make them separate from there, and in this way making the 100% DNA based complementary strand in case of the DNA replication. So second one is also getting that specific points of having the DNA replication process. I hope so you got that points. And now the third step also we will try to understand, and we can go through here also the strand of leading strand and lagging strand of having that parts. First one is the leading strand. In case of these two parts, leading strand is just followed by first one. In any case, in case of the DNA replication, the leading strand always try to move forward and followed by the lagging strand also be there. So there will be leading strand, there will be continuous process. There will be the continuous process where there will be no chance of separating leading strand, which one is there is no chance of getting separation, no chance of getting disjunction. Always they are going through with continuous process. That means they always try to form up with the parental strand and get it the photocopy or like the duplication. There will be no change in there. But in case of the lagging strand, there will be non-continuous process. That means sometimes it continuity will be non-continuous process. That means sometimes the process of making that specific parts with not having the continuity, and we have seen there will be some part is detached, not forming the 100% photocopy. So leading strand always making sure that one the 100% okay, and lagging strand not maintaining the 100% of continuity in this way. Okay, and we can see there also in any other case we can see some strand with their parts, with the template and complementary strand getting some separation, some segmentation, and parts also getting, we can see that parts are known as the Okazaki strand or Okazaki segment, you can say. So these types also we can get on that parts. And in this way, when finally the leading strand and lagging strand are going on through the replication process, the process is going on in forward, the DNA polymerase enzyme and formed. So again, suppose they move on there, that binding part will be unbinding once again, and the following, which one is complete their replication, they again twist and bind. In this way, finally the parental DNA with having that one, we can see the similar kinds of photocopy like duplication of the parental DNA with the daughter DNA are match up there. In this way, total replication process is done. And also having DNA repairing, also we can get if there are any nitrogen base sequence change, so they are needed to repair, and proofreading also be there. So in total process is going on to the replication process. So I hope so what I actually mean for by doing the figure structure or we draw the figure and try to understand the figure. In this way we understood the DNA replication process. I hope so you understood the today's lesson also. If you have any confusion, please let me know in the comment box. So we will try to solve here. I can give you the correct one what you have facing the problem in this class. That's all about today. We will meet once again with a new topics in the next class. Till then, everyone stay safe, stay sound. Allah Hafiz.